The research for more effective cancer treatment has led scientists to explore innovative approaches in vaccine development. At the forefront of this work is Dr. Tadao Ohno, President and CEO of Cell Medicine Inc., and an honorary member of the American Society of Clinical Oncology, who developed this innovative cancer vaccine. In this exclusive interview, Dr. Tadao shares insight into the science behind the vaccine, its clinical results, and what it may mean for the future of cancer treatment. Welcome again, Dr. Tadao. Can we please get to know you a little bit?
You introduced me completely, so I don't want to add more.
We look forward to having this chat with you and introduce you to the world with this groundbreaking development.
Thank you very much.
Welcome, Dr. Tadao. It's a pleasure to have you here with us today to talk about your innovative approach to cancer treatment and cancer vaccines. I want to start with your story and your motivation in cancer vaccines. Therefore, my first question would be, how did this idea for this cancer vaccine first emerge?
Okay, let me answer. One of the key immune cells that can kill cancer cells is the cytotoxic T lymphocyte, or CTL. By the 1990s, researchers already knew how to induce CTLs from mice in culture, but it was widely believed that human CTLs couldn't be induced in the same way. At that time, people thought you need the live cancer cells as targets in order to generate CTLs successfully. I was studying how to induce human CTLs, and I found that CTLs could be generated against established cancer cell lines, which grow well in culture. But those cells, CTLs, couldn't be given back to patients because the cell lines come from other people's tumors. CTLs trained to attack someone else's cancer cells can't recognize a patient's own tumor as the enemy. For cancer patients, the ideal CTL target is their own tumor cells, which carry that individual's unique tumor antigen. We need a large number of those cells, but surprisingly, culturing patients' own tumor cells turned out to be difficult. So I began to wonder whether the tumor cells in pathology slides, cells that are completely dead due to formalin fixation and embedded in paraffin, might serve as a substitute for live tumor cells. When I tested this idea, CTL induction worked. That was in 1995. If CTLs could be induced using tumor cells that were formalin fixed and embedded in paraffin, then it should be also be possible to take the original tumor tissue before it becomes a pathology slide, cut into small fragments, add immune adjuvants, and return it to the patient body. The patient's immune system should then mount a cellular immune response against the tumor antigen. That is how the concept of the autologous formalin-fixed tumor vaccine, AFTV, was born.
Very good. Well, very nice explanation. Thank you. On to the second question. I want to ask you, how many years have you been actively working on this vaccine technology itself? And since then, have you had clinical applications been conducted?
Yes, I've been working on this technology for 32 years, starting in 1994. Clinical applications began in the early 2000s.
Very nice. How many clinical applications have you done since then?
The application began, in the early 2000s, around 2001 or 2.
Okay, very nice.
Published earliest one, 2003 to 4.
Very good. Mr. Tadao, you've mentioned all these amazing technologies that you've invested yourself into, and you have led research in this field for 32 years now at Riken, one of Japan's most prominent scientific institutions, often described as Japanese equivalent of TÜBİTAK in Turkey. How did this experience contribute to the development of cancer vaccine?
Riken had a very generous research fund, which allowed me to hire postdoctoral researchers and skilled technicians. Because of that, I was able to build a strong research team, and our work progressed efficiently.
Very nice. nice. And moving on with your story of this vaccine and your motivation, the approval of the cancer vaccine you developed is considered a major milestone in the medical world. How do you think this development will transform the future of cancer treatment itself?
AFTV is extremely safe with almost no significant side effects. However, it's not something that works immediately. It takes about 3 months before the effect appears. So it's a slow-acting therapy. For that reason, I see it as a complementary treatment to other therapies that works quickly, such as radiation or chemotherapy, which are effective but often come with strong side effects, as you know.
Yeah, very nice. I think it will evolve this entire medical world and the cancer journey of a patient. So in recent years, immunotherapies have gained significant importance in cancer treatment. So I want to ask you, in what ways does the vaccine you develop different from existing immunotherapy approaches?
Let me answer. Most current cancer immunotherapies are immune checkpoint inhibitors, but they can cause serious side effects such as interstitial pneumonia. With AFTV, we don't have that concern. Okay? Its high level of safety is one of its biggest advantages. All right.
Very nice. So on to my next question. How does your vaccine differ from other cancer vaccines known worldwide, such as a therapeutic cancer vaccine developed in Cuba or dendritic cell vaccine approach?
Okay. Cuba has developed cancer vaccines. These vaccines are not personalized for each patient. They are manufactured in the same way as conventional pharmaceuticals, and every patient receives the same product. They are designed as therapeutic cancer vaccines. In contrast, AFTV is made exclusively for the patient who underwent surgery. It is a truly personalized vaccine containing, at least in theory, all the antigens derived from that patient's own tumor. And importantly, it does not require cell culture. It contains no living cells at all. This vaccine is not intended to treat large tumors that remain in the body. Instead, it is particularly effective as a post-surgical preventive therapy, targeting microscopic residual tumor cells that could otherwise lead to recurrence. Dendritic cell vaccines are also personalized in the sense that they use the patient's own cells. However, they require extracting dendritic cells from the patient, culturing them outside the body, and loading them with tumor antigens during culture. Culturing live dendritic cells requires very careful management, which makes the process costly. All right?
I see. Perfect. Thank you for explaining that. Now I want to move to the second part of our interview, talking about the scientific foundation of your development. So my question goes, what is the scientific principle behind this vaccine? How does it activate the immune system against cancer cells?
Okay. In simple terms, if we work by enhancing the body's cellular immune response against cancer cells, a patient's own tumor cell, cancer cell, carry unique tumor antigen, including neoantigens that are not found in normal cells. When these antigens stimulate the immune system, they help expand powerful cytotoxic T lymphocytes or CTLs, which are capable of killing cancer cells.
So considering the immunosuppressive nature of the tumor microenvironment, how does your vaccine overcome this immune suppression?
This is a very difficult question to answer, but we rely on the action of the immune adjuvant that we add to AFTV. However, immune adjuvant alone may not be enough to fully overcome the immune suppressive tumor microenvironment. That's why we are currently studying combination approaches, particularly with radiation therapy or immune checkpoint inhibitors. All right?
I see. Okay. Based on your experience, how do you think nanotechnology-based approaches could play a role in cancer prevention or early prevention in the future?
Well, in the future, yes. especially for the development of new immune stimulants, such as we say adjuvant. I believe nanotechnology-based approaches will play an important role in that area.
Now I will move on to our third section, which is the clinical application, which I'm very curious to hear about. So how is the vaccine administered to the patient, and what does the clinical application process and the protocol look like?
Okay, but would you please proceed your question to the next one
Yes, we're skipping this question?
together with next question?
Yes, and I will ask the second question as well. My other question is, how many doses are administered during the treatment, and how are the dosing intervals determined?
Let me answer. AFTV is administered by intradermal injection. We give a total of 3 injections, usually every 2 weeks, that constitute one course of treatment. If we need to move faster, we can shorten the interval to 1 week or sometimes 10 days, but the total number of injections remains 3. In principle, one course is sufficient. However, if a patient strongly wishes to receive more than one course, and if we have enough tumor tissue available, we may repeat the course 2 or 3 times. The protocol itself is very simple.
I see.
Alright.
And how is the treatment plan developed for a patient? Is it a personalized approach?
We still studying which patient conditions most strongly influence the effectiveness of AFTV, but at minimum, there are several situations where AFTV cannot be used. First, patients who already have autoimmune diseases can't receive AFTV. The immune stimulation from the vaccine is very strong, and it could rapidly worsen their autoimmune condition. Second, patients with very advanced cancer who are already in a terminal cachectic state, unable to mount a cellular immune response. In such cases, AFTV would not be effective. Third, patients who have received prolonged cytotoxic chemotherapy for solid tumors may have severely damaged bone marrow. If the bone marrow can no longer produce young lymphocytes, the precursor of killer T cells, then AFTV cannot work because the immune system cannot respond. Long-term steroid use will also have damaged young CTL precursor cells. Finally, cancer like leukemia, where there is no solid tumor mass to remove, are also difficult to treat with AFTV.
I see.
All right.
On to my next question about the clinical application. It would be, can this vaccine be used in combination with chemotherapy, radiotherapy, or any other immunotherapy together?
Yes, it can be combined with chemotherapy as long as the chemotherapy is mild enough or given at a low enough dose that it doesn't harm young, healthy lymphocytes. If the treatment does not damage those lymphocytes, then combining it with AFTV is not a problem. Combination with radiation therapy is actually quite favorable. When cancer cells are destroyed by radiation, they release various substances that can act as immune stimulants. So the two treatments, AFTV and radiation, can complement each other well. AFTV acts as the accelerator of the immune response. Immune check inhibitors, on the other hand, release the brakes of the immune response. Because of this accelerator on break off relationship, we expect that combining AFTV with checkpoint inhibitors may produce synergistic effects.
Amazing.
All right.
Thank you. So which patients are considered suitable candidates for this vaccine? I know you mentioned a little bit, but I want you to tell me more. And are there specific biomarkers, genetic characteristics, or clinical criteria?
Well, at present, we do not yet have specific biomarkers or genetic characteristics that predict who will benefit most. However, we do have 7 practical criteria that indicate whether a patient is a good candidate for AFTV therapy. First, we need to obtain 3 to 4 paraffin-embedded tissue blocks from the patient's surgery and total amount of tumor tissue inside those blocks should be estimated at 2 grams or more based on microscopic photograph with a ruler, just like this. This assessment will be performed by cell medicine in Japan by us. Okay? Second, the patient must be able to send us their most recent CT images on a CD CD-ROM by air courier before leaving Turkey. Japanese physicians use these images to determine eligibility for FTB therapy. Third, the patient must not have any autoimmune disease. This requires documentation from the patient's primary physician in Turkey. Fourth, the patient's peripheral blood lymphocyte count should ideally be 1,000 per microliter or higher before departure from Turkey. This should be confirmed by the local physician's laboratory report. Fifth, the patient should have a Karnofsky performance status of 90 or higher, meaning they have mild symptoms of cancer, but can carry out normal activities, and the expected survival should be at least 6 months, again documented by the local physician. 6. The patient must be able to walk independently without a wheelchair, tolerate round-trip travel from Turkey to Japan, or it need 11 hours or more, and stay at a hotel near treatment clinic in Tokyo during the therapy period. That also should be confirmed by the local physician. And finally, it is preferable if the patient is well enough to enjoy 2 weeks of sightseeing in Japan during the intervals between AFTB treatment. All right?
Amazing. Okay. So, talking after the 7 criteria, at which stage of cancer does the vaccine show highest level of effectiveness? And do you observe different outcomes between early stage and advanced stage patients?
We have not yet completed detailed analysis by cancer type and stage. However, in breast cancer, as I have presented at CyberCellX Future 2026, patients who developed distant metastasis stages after surgery, all of whom were stage 4, showed the longest survival reported worldwide when measured from the day their metastasis was detected. With radiotherapy in combination, their 10-year survival rate exceeded 50%, which as of March 2026 is the highest survival rate in the world for this patient group.
So which type of cancer have shown effective results with this vaccine so far based on your tests and development stages?
Okay, the first clear result was in liver cancer, where we demonstrated a post-operative recurrence prevention effect. We published those findings in 2004. Next, we have seen promising result in breast cancer. Currently, we are conducting studies to see whether the vaccine may also be effective against glioblastoma, a type of brain tumor.
Wow, we wish you the best of luck in this. This is a miracle for us. Now I want to move on to our fourth part, which is the clinical outcomes. Moving on to the clinical outcomes, I'd like to ask you, what have been the most remarkable findings from your clinical trials?
The most remarkable finding is the post-operative recurrence prevention effect in liver cancer. This was demonstrated in a phase 2 randomized controlled clinical trial, where patients who received AFTB were compared with randomly selected control patients.
And what is the success rate of these clinical outcomes?
Okay, in liver cancer, AFTB reduced the relative risk of post-operative recurrence by 81%. It's amazing. Even the highest risk group, patients whose cancers were 5 centimeters or larger in diameter, where the recurrence rate is typically 91%, the reduction in recurrence was statistically significant. When I presented these results at a Japanese conference in 2002, the effect was so striking that almost no one believed it at the time.
It is still striking. Amazing. Congratulations again. Moving on to my next question. How effective has this treatment been in improving patient survival? And what does the clinical data indicate?
In the phase 2 randomized controlled trial, at 24 months of follow-up, the survival rate was 19% in the AFT group, while 50% in the control group. This difference was statistically significant.
Very much.
All right.
Very much. And could you share any striking patient cases with us that have benefited from this treatment?
In that particular clinical trial, we were not allowed to report individual patient data. However, outside the trial, there was a memorable case of a patient who developed liver cancer due to hepatitis C. After the first surgery, the cancer recurred almost every year despite undergoing 28 different treatments, including a second surgery. The cancer recurred again, and the tumor marker AFP, well-known alpha fetoprotein, eventually rose to more than 40,000 nanogram per ml. After the third surgery, this patient received FTV. Following that, the cancer did not recur for more than 5 years, and during that entire period, the patient received no other treatment besides the FTV. This is very much important point, all right?
This is a very remarkable story you shared with us. Thank you. Now I would like to move on to our fifth stage, which is the safety and follow-up after the treatment. That being said, I would like to ask you, what can you tell us about the safety profile of the treatment and what side effects have been observed in these clinical trials?
There is an international standard called CTCAE, which is used worldwide to evaluate the safety of anti-cancer treatment. In this system, grade 3 and grade 4 events are considered serious adverse events, and if it is grade 5, indicates patient death. In the case of AFTV, whether in liver cancer trials or breast cancer treatment, all adverse events were only grade 1 or grade 2, the kinds of reactions that resolve naturally even without treatment. We have never encountered any serious or problematic adverse events. All right. And even among the 59 breast cancer patients who received FTV after developing distant metastasis, as I presented at CyberCellX Future 2026, not a single patient experienced a grade 3 or higher adverse event.
This is a very successful rate. And after the vaccine is administered to the patient, how does the patient's follow-up process proceed, and how long does the immune response last?
Unlike the former clinical trial we conducted for liver cancer, the AFTV treatments currently provided in Japan are not part of a regulated clinical trial. This means we do not impose any requirements on patients, such as prohibiting other treatments or obligating them to return to the clinic every few months. We simply provide AFTV based on the patient's own wishes. Because of this, many patients do not return for follow-up after completing AFTV treatment. As a result, our follow-up data is unfortunately quite incomplete. Even so, in several cases, we have seen patients who 5 years after the initial FTV treatment still show a clear delayed type hypersensitivity reaction when exposed to their own tumor tissue fragment without the immune adjuvant. This reaction is evidence that a cellular immune response against their tumor antigen is still present. So we can say with confidence that the immune response lasts for more than 5 years, and theoretically, as long as the memory T cells responsible for this response remain alive, the cellular immune response to the patient's tumor antigen could persist for a lifetime.
In the long term, is there a possibility that tumors could develop resistance to this vaccine? Have you observed any signals suggesting such a mechanism?
Yes, that possibly does exist. As cancer cells continue to proliferate inside the body, they accumulate genetic mutations. These mutations can make the cancer progressively more malignant and more capable of rapid growth. If over time, cancer cells acquire new antigens through genetic mutations, antigens that FTV can't recognize, then those cancer cells may escape immune detection and the vaccine will no longer be effective. This is likely the mechanism behind the cases where FTV initially works well, but the cancer eventually recurs after some time has passed. Because our follow-up data is incomplete, we cannot quantify exactly what percentage of patients experience this. However, we have seen a considerable number of such cases.
On to our sixth part, which is the global outlook. And I would like to talk about the global access and collaboration. And my question follows as, what are your plans for making this treatment accessible to patients worldwide?
AFTV is still an unapproved medicine at the national level. In Japan and Germany, physicians are allowed to use unapproved treatments under responsibility. But as far as we know, most other countries do not permit the use of unapproved drugs in cancer treatment. For this reason, patients from Turkey currently needed to travel to Japan to receive AFTV. In the future, we hope to make treatment available in Germany as well.
Amazing. So when do you expect widespread use to begin?
In Japan, we already collaborate with more than 80 clinics that are permitted to use unapproved treatment for cancer care. We plan to establish similar partners in Germany in the future. At the same time, in Japan, we are conducting formal clinical trial to obtain national approval. Because development costs are extremely high, we are currently conducting trials only for brain tumors. But in the future, we hope to run official trials for other cancer types as well.
In doing so, how do you evaluate your collaboration with Turkey, and what position does Turkey hold in this field?
Since AFTV is still unapproved in Turkey, it cannot be used for cancer treatment there. Therefore, for Turkish patients, the only option we can offer at the moment is to introduce them to treatment abroad if they wish to receive AFTV. We have high expectation for Turkish companies involved in medical tourism, and we hope they will play an important role in connecting patients with these treatments.
Amazing. And then now I would like to move on to our last section, which is personal reflections and the future vision in this development. Following the development of this vaccine, what is your next scientific objective?
Going forward, I would like to focus on how to make AFTV even more effective by combining it with other treatment modalities. Developing optimal combination strategies will be an important part of our coming research.
Amazing. And then looking ahead, 10 to 20 years from now, what kind of transformation do you foresee in cancer treatment?
Well, AFTV can only be used for patients who have undergone surgery, but I believe that the core concept behind AFTV, namely cancer immunotherapy, will expand to benefit patients who cannot undergo surgery or who do not need surgery. I expect immunotherapy to become increasingly central across many stages and types of cancer.
Amazing. And what advice would you give to young scientists who aspire to work in the field of cancer research?
Okay, cancer is a disease that humans will inevitably face as long as we live. The challenge of finding better ways to control cancer will never disappear. My advice is commit yourself with the determination to keep working steadily and persistently without giving up.
Amazing. And finally, Dr. Tadao, it's been amazing having a chat with you, but what is the most important message you would like to share today with patients and families who are fighting cancer?
Okay, let me say, even if it is not possible to eliminate all the cancer in the body right now, please hold on to the hope that as long as the disease does not worsen, you can continue living your life. And if you can keep going, new and better cancer treatment will surely emerge in the future.
Thank you for sharing that message. Dr. Tadao, thank you very much for joining us today. And also, thank you for taking the time to explain this groundbreaking work and the development that you have led for the past 32 years. Your development of this cancer vaccine marks a significant breakthrough, and we're grateful that you have shared your insights and your vision with our audience and patients and the practitioners in this field. Thank you so much.
You're welcome. Thank you very much.